term why. So this is, yeah. Okay, recording in progress. So digital sequence information is a term used in policy circles for data derived from genetic resources. And genetic resources is a specific thing within the context of regulation for access and benefit sharing. And so digital sequence information today, when I talk about it, I'm mostly going to be talking about genetic data, but it's actually more complex than that. The Nagoya Protocol is the international regulatory framework. It's a supplement to the Convention on Biological Diversity, and it regulates the sharing of benefits from genetic resources, the physical genetic resources. Again, I'm oversimplifying. And so you'll hear that come up. So people who use physical resources from around the world are used to dealing with this. And then there are the parties to the Convention on Biological Diversity are currently negotiating a new benefit sharing policy measure for DSI as part of their new action plan, the Global Biodiversity Framework. Okay, and why does this matter to you as a researcher? Well, this has turned out to be a really crucial part of these negotiations on the Convention on Biological Diversity. The outcome of this could have a dramatic impact both on how benefits are shared from molecular biology, genetic research, but also how you as a researcher are able to interact with genetic sequence. We all take for granted that we can just go and download the sequence that we need from open databases. And I think that there are powerful advantages to that, but that is what it's at stake now in the current policy discussions. There we go. Skip over this very quickly. Today, I'm mostly talking about DNA, but you're going to hear that digital sequence information is a broad term specifically, and to scientists that can feel kind of pointlessly fuzzy. But in policy circles, it's really important because it's actually part of the policy discussion. Where do you draw these lines? And we will be hearing about traditional knowledge today. And so why is this now on the international radar? Well, when Nagoya was negotiated in the early 2000s, since that time, there has been this explosion of sequence data in the public repositories. And of course, that exponential curve started already in the late 80s and 90s. But increasingly, there's this recognition by parties that says actually a lot of the value from genetic research is derived from these data. And some people feel, parties feel that they are not getting the benefits they deserve out of this system. On the other hand, there are a lot of researchers that really understand the power of this open data sharing. So DSI use on a global scale is complex, collaborative, and global. The whole data set in the main sequence repositories is downloaded over 30 million times per year. And this is not something you can just do on your laptop, right? So this is a specialist task, and it still happens millions of times. There are easily over 100 million unique users based on the web stats from these databases, probably many more. There are thousands of interconnected databases which share data back and forth, and there's at least 1,000 that have been documented in previous reports. And there are hundreds of thousands of publications that cite many sequences in them. It's complex. DSI are also shared openly by researchers. I'm not really going to talk about this much today, but I want to introduce that briefly, is that when people talk about open, open has a specific history within scientific publishing and a specific history within code sharing, for example. For genetic information, open generally means, and these principles are derived from these two papers down here that Rodrigo is the lead author on both of those, but you'll hear from him later today. So access is anonymous, free of charge, and free of restrictions. And DSI are shared in ways that are interoperable and support scientific transparency and reproducibility. So the policy debate really comes down to how do we continue to preserve these important principles? Which of these principles are most important? And how do we do that in a way that then more effectively shares benefits back to the countries who are preserving their biodiversity? Now, I'm going to talk, so that was my intro. I'm going to talk about three things, and I'm going to maybe skip over a few slides to try to keep us on schedule. So first, I'm going to talk about some data-driven work that we've been doing in our group, and I'm only going to introduce this briefly because there's a cool web portal that you can use. We've been looking at data to try to understand how we can really see global patterns of digital sequence information sharing and what that tells us about policy. We've been looking very briefly at patents, which I will probably do just a taste of. And then I want to introduce at the end why we feel, based on this work that we've been doing in the group, why we need an effective, fair, and open multilateral policy solution, and why you as researchers should be raising your voice for that. So within policy circles, there is a really common narrative, and I think it's important for researchers to speak up, but we've been trying to use data to approach these narrative. So this common narrative is that biodiverse, economically developing nations provide DSI to economically developed user nations, so that there are providers and there are users. And anyone who's been on this policy topic for a long time knows that this is an oversimplification, but you continue to hear this in negotiations. It continues to be part of the drive within the negotiations. And so it may end up affecting the final outcome policy solution. So we've been using two data sources. We've been looking at sequences from the major repositories and the open access literature. And with that, we can actually, here's an example of a paper. So we're using text mining. We can say, okay, this paper, we can find the corresponding author, we can find the country. So we say that this is a user from Peru. We can then go in the paper and we can find citations to sequences. Okay, you can see this AF307. We can go into the public databases using structured database calls. And we can find the country of origin, which is Brazil. And this is actually not where the sequencing happened. This is where the biological sample originated, which is what matters under Nagoya. So we can say that we have a user in Peru and a provider in Brazil. And using these data in bulk from looking at millions of papers and millions of sequences, we can start building a global view of what's actually happening. And so I'm going to show you two very quick examples. So this is looking at the top providers and users of DSI. And blue is DSI originating from a country. Purple, the middle bar, is DSI using publications. So how much are the researchers in that country using DSI based on their publications? And then the red bar, how much is the DSI from that country being used by other researchers? You don't need to understand the bars, but what you see immediately is that while there's some real weird outliers there, like Canada, overall, there's this strong correlation between use and providing. There's not one group of countries that are providers and one group of countries that are users. And you can also see that there are a lot of sort of middle range economically developing countries that are in this top 25, right? So it's a complex picture. You can see this, again, we can address this myth more directly. This is we have a way of sort of measuring how much a country is using DSI and how much a country is providing DSI. Here we're actually measuring as a proxy the number of countries that are using the DSI from that country. Again, it's a bit complex, but what you can see is that there's this really strong correlation on a national level between use and provision. And if there were just provider countries, right, you would expect them to be up here. And you can also see that economically developing countries, the G77, they often tend to start out, well, we're looking at this from a temporal point of view in our current research, but there seems to be this progression where you start out by using DSI and then you grow into being more of a provider. So anyways, we're using these data to sort of challenge these common narratives, these common myths, and to remind researchers how much researchers in economically developing countries depend on sequence from elsewhere. We have ways of filtering this data now to look at other sorts of policy for us. So we can look at just plant. So within the plant, this is just proof of principle. If you just look at the plant data, you can see that China is the largest international provider compared to all digital sequence information. We can do this for the oceans. You can see that plants are the largest proportion when you look at the whole data set. And when you look at oceans, no country, which is going to be mostly non-territorial waters, this mostly deep water, there's a lot less plants. Okay, proof of principle just to show that there are interesting patterns in these databases if you're looking at different policy contexts for example, the plant-specific treaty, which I haven't talked about. On that topic, this is one of my most important slides, although not super exciting unless you're into this. So there's a separate treaty that regulates access and benefit sharing for crops. And this is called the ITPGRFA, International Treaty on Plant Genetic Resources for Food and Agriculture. All right. And Annex 1 lists the crops species that are covered by this. And we've been transforming this into a machine readable file. It's actually quite a somewhat complex taxonomic task, just so that we can do filtering automatically. So for other people that are doing databases and have to do compliance with Annex 1, this could be a useful resource. If you happen to be a taxonomist, we're now at the stage where we are looking for taxonomists to help validate our mapping. So get in contact with us. So all of what I've just talked about has been work of others. I've been involved in it, but only at a late stage. And it's been done in partnership with the group at the IPK. Most recently, Matthias Lange, Jens Freitag, and Jorge Garcia, and with a lot of work from EMBL EBI. There's a portal there at the bottom, 2 papers where you can learn more. So I'm going to go, because of time, very quickly over the patent. So people say, fine. Academic publications, fine, it's complex, but what we really care about is value in patents. So we've been trying to use data to look at this, how are DSI being used in patents? We are building a text mining infrastructure to look at this. This slide shows you nothing other than we can start generating some data about how many sequences are connected to patents. So we're also looking at individual patents, and the story that we see, this is a great story, which takes a whole 10 minutes to tell, so you're going to get the really, really short version. The Moderna patent is based, the final vaccine is a single sequence. So everyone says, okay, it should be really easy to regulate. But when you look at the actual patent, it never mentions a single SARS-CoV-2 sequence. It actually mentions hundreds of other sequences, and assessing which is the valid country of origin is almost scientifically impossible. So this idea that for a product that in the end is a single sequence is actually much more complex. So for time, I'm going to go over that. I'm going to skip over that. And we're going to go to the very end, which is our take-home message. So we believe that there are policy arguments out there that would either like to control sequences like genetic resources. So to get an individual sequence, you would have to track which individual sequences you're using, fill out paperwork, blah, 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 and pay agreements. We believe that that would be ineffective because there are real research is not about individual sequences, it's about thousands or millions of sequences all at once. For this to be effective, scientists need to be able to share their data openly. And for that to happen, we need multilateral policy solutions. Multilateral means that you don't have to understand the laws of hundreds of countries in order to do your research. We have authored a proposal with a group of researchers, which you can read at Nature Communications, which lays out the broad principles of such a multilateral system. This has been authored in cooperation with the DSI Scientific Network, which is an international network of researchers who are concerned about this issue and are trying to raise the voice of the research community. This is the figure of the proposal. The basic idea is that you're not charging for access to individual sequences. Instead, there's a multilateral fund that some parties are paid into. Payment requirement is triggered by using any sequences at all, and then payments come out of that system based on a project basis. Does it preserve biodiversity? And to some degree, you link that to whether countries are sharing their data openly. So you create an incentive for biodiversity research, you create an incentive for countries to share their data, and real benefits flow back to them if they do it. Now, of course, there's a lot of particulars in that, but we think that this is the outline of what the research community needs. There's an open letter now signed by over 1,000 individual researchers and more than 32 international scientific organizations. Link there at the bottom, dsiscientificnetwork.org/open letter. I encourage you to read it, sign it, and think about how this works with your own research. Talk to your funders. So thank you very much. only a little bit over time. Big thanks to, so I'm Andrew Hufton. My boss is Amber Schultz. She's the head of the science policy group. And so all of what I've talked about is work led from there. Credit to Rodrigo Serra, because he's here today, and I've talked a bit about some of his work with that group. And Scarlett Sett, I didn't mention her earlier, but she works closely on some of our Nagoya-related and environmental virus archive work and has helped contribute to some of these slides.
Thanks, Andrew, for really. So we've got microphone. Thank you for clawing back some time for that. So we really appreciate it. It'd be a great story, but we'll have to leave it for another time. But I think we should give you allow time for question. So has anyone got any questions at this stage? Yes, one here. Yep.
Okay, so I was wondering, like, what the framework a few slides back near the end that you propose was a multilateral system rather than similar to the current system for material transfer. I was wondering, do you think that this system for digital sequence information could be used as an example to essentially also suggest that the material transfer should also work in a similar way? So it's not about an individual permit and agreement tied to a sample, but it's about open access in exchange for a general sharing in benefits that aren't tied just to an individual project, which, well, also for the provider countries in a given case may not actually make sense if you just like do similar research for some other country instead.
So that's a very good question. And so to repeat to the room, you're saying, well, why are we building a new system for DSI? Could this be, if this is such a good idea, could this be used as a framework for a better way for physical samples, right? And so the answer to that, like, that is a very reasonable proposal. Politically, it's complex. So I think the business world is specifically proposing, we don't want a DSI solution at all until Nagoya is fixed. And I understand their position on that, but it is risking blocking the whole biodiversity treaty. On the other hand, there's the plant treaty, which I'm not an expert on. I know very little about it. And the plant treaty, I think, is a template for a multilateral system for sharing physical samples. And so this DSI treaty, if it is layered on top of, it's going to be layered both on top of the Nagoya, which is bilateral, and the plant treaty in a relatively complicated way. And hopefully that will, everyone wants consistency because in the long run, and not several different instruments. And Africa is specifically proposing a broad tax on all biodiversity related products, which is an interesting proposal for various reasons, but we don't have time to get into it. Yeah.
Thanks very much.
Yeah, we definitely have time for one more question.
Yes, I've got a question and a comment from the framework that you presented and the open letter. I'm going to sound quite negative. Just a bit of a, what I see as a flow, it's been designed and developed by scientists. And I don't like using the term Western scientists, but that's the fact. So it needs to be co-designed by indigenous people. And that's what's missing here. There's some shortcuts that are done, for example, saying that, countries, could, give some resource to maintain this framework, but often indigenous people doesn't equal countries. There's often a very strong divide between indigenous people who have been actually colonized and screwed by their government. So it's not actually, I'm sorry for being negative, but I'm all for, policies and framework that work for everyone. But in this case, it's not working for everyone. There's actually a massive amount of people missing from this framework, and that's actually a co-design from actual indigenous people who miss out systematically from those type of well thought framework. So
yeah, I won't try to disagree with that. As I said, I've only been working in this space for about a year, so I cede to the greater experience of many people in this room. But what I will say is that the proposal there was a great effort when drafting the Nature Communications paper to take into account the perspectives of researchers from around the globe. No, and I emphasize that on researchers. And so I think that there is this idea that somehow researchers in economically developing countries want bilateral is not true. There are a lot of researchers, most researchers around the globe want a system that allows them to continue to do work. Now, I think the open letter, the open letter does acknowledge that indigenous peoples need to be involved. They have been at the table, but it is a fraught discussion. I'm not sure what the solution is there. I know that traditional knowledge, we're going to hear more about it today, and that's being discussed in other forums. I think that this is obviously something that's going to need to be solved, and that sovereign-based negotiations are a difficult way for that solution to come out. But I don't think that making the system closed and trying to regulate individual sequences personally is the solution to that.
It's not all about being closed, but it's about getting the right consent and agreement and control from those communities. And I mean, if your system can achieve that, fine. But it doesn't seem to me at the moment, and tell me if I'm wrong, that it's been co-designed and co-thought by communities.
What's going to come out of this, obviously, is that I think this is a solution to maintain the existing public system, which everyone would like to maintain. There are already sequences that are held in controlled access repositories of different kinds. And I think that there's a discussion. So I'm of the view that we shouldn't be trying to move the public repositories into controlled access systems. But I think we do need to have systems in the end that create controlled access systems for special types of sequences. I don't think that people will be able to generate benefits off of those kinds of systems very effectively, personally. But that's a challenge, and I think we're going to have to experiment with all of those systems to make it work. Because sometimes it's not just about generating monetary benefits; it's about respect and control, right? Yeah.
Just before we go on, I just want to find out where we're at with the next speaker. How are we going? So Brad's not in the room yet. So seeing Brad's not in the room, we can just joined.
I am.
Oh, hi, Brad. That's great.
Yes.
So we'll put you on. What I'm going to do is, does that mean stop sharing the page? Yes, if I click that. Okay. So Brad, I'm going to stop sharing the page, and then can you put your sides up and share yours? He's putting his coat
sure.
on. It's good. It's a nice coat. It's obviously winter.
It's freezing.
So I'm going to close this page, and then you can share yours. Okay. And I'll just introduce you while you do that too. So the next, thank you. So first of all, thank you very much for Andrew for his talk. And I think it just shows you that we're all on the same page and we're having this discussion. So how complicated is it when we even get into a wider community? So now it's my pleasure to introduce Brad Sherman. Brad is a professor of law at the University of Queensland. He was a co-author with me and a professor of crop science there on that plant scientists need to urgently consider these matters to know the way forward. So he's joining us online from where it's very late. I need to make him full screen somehow. I mean, I can do it in English. I'll let you do it. I'm too scared. Okay, so I'll hand over to you, Brad, and if you can share your screen, then we can see it.
Okay. Can you see that yet? Can you see my screen?
We can see your Outlook page.
Okay, sure, good.
I'll make sure we edit that up.
Yeah.
Okay, that's great.
It's okay.
Yep. So you just need to, yep, there we go. Great, thank you. Well, all systems go.
Okay, thank you. I'll just close it down.
Yep, no, it's good.
Okay, can you hear me okay?
Yep, yes, we do. Yep.
Okay. Okay, thank you everybody, and sorry I missed the first talk. The discussion tonight will be on looking at access to genetic resources under the Nagoya Protocol, and I'm focusing specifically on the problem historical collections. So as you're probably all aware, the Nagoya Protocol was passed in 2014 as a result of disquiet about the effectiveness of the CBD by member countries. Its goal was to provide guidance about how the CBD was to be implemented. And while the parties could agree on many points, a number of issues were left open to different interpretations when it was passed. The Nagoya is an incredibly important treaty for a number of reasons. Probably the most important is that it regulates behaviour outside of the countries where it's been adopted into national law. So this means, for example, although, say, the United States or is not a member of the CBD, or Australia has not yet implemented the Nagoya, it still regulates behaviour in Australia. The reason for that being is that when someone from Australia exports to a Nagoya compliant country, the Australian researchers will need to show that they have acted in a Nagoya compliant fashion. The second reason why Nagoya is important is because there's a growing number of non-state actors, such as universities, herbaria, scientific journals, which have made Nagoya compliance a precondition of dealing with biological materials. So, for example, the University of California, so as I just mentioned, the United States is not a member of Nagoya. University of California has decided unilaterally to be Nagoya compliant. So an Australian researcher who wanted to send materials to California to be sequenced was told that they would only deal with that material if he could show that he had collected it in a Nagoya compliant fashion. So there's a growing number of institutions that are thinking about doing this. So the question, who needs to comply with Nagoya given this scenario? So obviously researchers in countries that have adopted Nagoya, and there's 120 plus countries that have adopted it, they would need to comply with local domestic laws. So people in France would need to adopt the EU model, people in South Africa with the South African law, et cetera, et cetera. Researchers who want to export genetic resources to a country that has adopted Nagoya must comply with the law in that country. So an Australian researcher who wants to export to France would have to ensure that they comply with French law in other countries as well. And then thirdly, researchers who want to work with non-state actors, such as the University of California, that they decide they have to be Nagoya compliant. So this means, as I just said, an Australian who wants to export material to France must show that the material is Nagoya compliant, or an Australian who wants to send material to the University of California would need to show the material is Nagoya compliant. Now, what does it mean to be Nagoya compliant? It's pretty straightforward. You need to show that you have prior informed consent and that there is access and benefit sharing, which is obviously monetary and non-monetary benefits as well. In most cases, that's pretty straightforward. However, a number of problems arise, and I want to focus on one of those in particular, and that's in relation to historical collections. The reason why this has arisen because there is uncertainty in the Nagoya about its temporal scope. During the negotiations leading up to the treaty, there was a number of different approaches, but there are two different competing approaches. One approach, it was from developing countries primarily, was arguing that the treaty should be retrospective, so it should apply to whenever material is collected. Developed countries, EU, Australia, and other countries argued that it shouldn't be, it only should apply once the treaty was in force in that country. The parties couldn't reach agreement, and it is left open, and as a consequence, different countries have had the opportunity and have taken up the capacity to adopt different approaches. So in Europe, for example, the Nagoya Protocol is limited to the time after the European member and its 28 member states implemented the treaty. So if someone's using historical material in the EU, or if they're a non-EU country wanting to export it to one of the 28 EU member states, as this is historical material, as it falls outside of that period of time, would not be necessary for them to show that the material is Nagoya compliant. In some countries, and there's a growing number of countries, however, South Africa, Norway, two examples, The protocol is triggered whenever genetic material is utilised, and they have interpreted that to mean this applies to material collected before the protocol or indeed the CBD came into operation. Many other countries are considering adopting this approach. This retrospective application creates particular problems for historical collections. The reason with that is, in many cases, the provenance of the material is unknown or unclear. There may be no information about where and when the material was collected, who the access provider was, or whether it was collected with permission, prior informed consent, benefit sharing, etc. It's also unclear whether the right to negotiate a retrospective access of benefit sharing agreement transfers with the land according to the access provider's will or some other means. So what would happen, for example, if the material was collected 100 years ago and the owner subsequently sells the land? People were not thinking about this, so there was no consideration given to this in succession. And as a consequence, it's incredibly difficult to know who the person is you should seek approval from going forward. This has created real problems with historical collections. Researchers working with historical collections may find they're unable to negotiate with relevant access provider to obtain the necessary approval. They may not even be able to, in many cases, find out who the relevant access provider is, and they may not be able to show that they are Nagoya compliant. So where does this leave researchers working with historical collections? So where an Australian researcher wants to export plant material, they'll be governed by the law of the importing country. So Australian-based researcher who wants to export historical material to France will be okay, but an Australian researcher, or in fact any researcher who wanted to export material to South Africa would not. Where a researcher is working with a non-state actor, such as the University of California, Journal of Molecular Ecology, and other journals, this will depend on what the non-state actor means by Nagoya compliance. My research to date suggests that while institutions have been thinking about becoming Nagoya compliant, they haven't gone one step further and explained what that means, other than access and benefit sharing and prior informed consent. It's unclear whether the Nagoya compliance in these contexts are prospective or retrospective, and most are silent on the issue, so therefore the result is unclear. The upshot of this is there's confusion and uncertainty. We've been doing work for about the last year or so in Australia. Every herbarium in Australia has every herbarium in Australia over the last year has restricted access to materials. They are making decisions not to collect, so they're not taking new accessions, and it's limiting research. I've started to do some work with botanical gardens and herbaria in Cyprus, and they're experiencing exactly the same problem. Anecdotal evidence from around the world suggests that these problems are happening in lots of different scenarios. The upshot is that this confusion and uncertainty is limiting access to research, and with all the consequential problems. Now, what can be done? And this is really the main point about my talk today, to just begin the discussions about thinking about what could be done. One option would be to reform Nagoya or adopt a new version of Nagoya going forward. International consensus is highly unlikely. The international treaty-making world now is incredibly fractured. 20 years ago, it was possible for developed countries to really push through treaties, and developing countries would just comply and go along with things. Things have changed considerably. The debates at the CBD about digital sequence information and just another example of a situation where it's highly unlikely there'll ever be international consensus just because of the divided nature of the sort of the different parties involved. One thing that I, along with other people, have been working on is the idea of thinking about developing best practice. So rather than waiting for reform at the international level, which is highly unlikely, this is an option that will build upon micro-level collaborative interaction and partner engagement at the lower level. There are a number of examples where this has happened, just two ones that might be relevant. When type specimens were first demanded by botanical, sort of zoological, taxonomic organizations, one of the conditions was that they obviously publish in the sort of the journal, the preferred journal, the details about the sort of what the new genetic material is going to be called, where it was found, etc., etc. They'd also list where the type specimen was defined, where it was deposited. One of the potential problems that arose in the 1930s and 40s was that people were taking out patents. This is particularly in relation to microorganisms, but also there's some push in relation to plants as well. People were thinking and trying to take out patent protection over the type specimen, which went against the spirit of the type specimen and its function and role in taxonomic and nomenclatural practices. In response to that, journals, a number of different journals made a condition of publication that the author would commit or promise that they had never taken out patent over the type specimen and they would never do so in the future. It was a condition of publication, much like ethics is sort of things like that now going forward. Similarly, in the 1915 and 1916, in response to particular problems with access to genetic, sort of access to publications and materials, publishers and royal societies, as they were known at the time, got together and developed what was called quaintly a gentleman's agreement. And this is an agreement where readers of journals were permitted to take one copy of articles by hand, obviously, and then circulate that to other people going forward. And it was the foundation for the library copying provisions which underpin a lot of public access to science today. So these best practice things have happened in the past, and this is the model and consensus of what we're sort of developed going forward. So what would best practice look like? So the idea here is that research organizations, universities, journals, herbarium, seed banks, funding bodies, etc., could clarify what they mean by Nagoya compliance via a best practice agreement. And obviously the best one would be one where there's consensus between these different organizations. And what I wanted to talk about today was really what these principles might look like and some of the issues that we can think about going forward. So the issues that we've been thinking about so far, we think that the best practice principles obviously should be consistent, easy to apply and uniform. They should recognise the role of access providers, even if they can't be found, and obviously the role of Indigenous peoples. They should facilitate scientific access and use. And at the same time, they should recognise the global nature of plant research, not just plant research, but all genetic research, means that researchers may eventually have to meet the higher standard of retrospective Nagoya, which is akin to the lowest common denominator. The reality is when people are starting off their research, they often don't know where the results will end up. And the idea behind this treaty, this best practice would be that people could, in this situation, and the best thing to do with them to ensure that they can get as broad of access as possible. One of the things we're thinking of developing is, in this context, is building upon copyright law's notion of orphan work. In copyright law, one of the problems that has existed for some time now is that because of the longevity of the duration of the rights, it's life plus 50 or life plus 70 now years, people may want, for example, to take a photograph and reproduce it in a book of something that was taken, the photograph might have been taken 100 years ago. The photographer is unknown or is dead or most likely dead, and it's unclear who they passed their copyright to, so who the members of their estate. In that situation, there's really two options. One is that you publish and risk infringing, and which publishers don't like and won't let happen. The alternative is you don't publish, and that's also what happens a lot. In order to ensure that this uncertainty about who the copyright owner was countries around the world, this is a European initiative initially, introduced the notion of orphan work. And this is a regime whereby if a potential user undergoes certain reasonable attempts to find the owner of the copyright, say in the photograph or the publication that they want to reuse, and they can't find them, they then have the capacity to use that work. And there's different options that exist in different countries. Usually it requires the user to contact some sort of collecting body or some sort of statutory body, who will then gives permission to the person, so gives them a license for them to use the material going forward. And in the event that the copyright owner comes forward, is located, arrangements are made to provide materials for them. One of the things that we have been working on as a part of the group of people who have been thinking is what an orphan plant might look like, because this is effectively what happens with historical collections. And we're trying to think about how we can develop an orphan plant regime that could be consistent, could recognise access providers and facilitate access going forward. What we are doing in this line is thinking about things like, for example, it might be the case that someone might want to get access to, say, a collection of macadamia that are planted in New South Wales. No, and it's unclear who collected them. It's certainly unclear who the access provider was, and then certainly it wasn't the case there's prior informed consent by the collector. So it'd be difficult for someone now to get access to those materials if you're adopting the lowest common denominator for Nagoya, which is something we think should happen for them to be Nagoya compliant. What we're suggesting is that the best practice models could make some sort of principle of which people could say, we have undertaken best efforts to locate the owner, the access provider of these provisions. We cannot find them. We are making a public commitment to share the benefits of the research, and it could be as simple as providing open access to all the publications. It could be sharing the materials or something like that. In the event that there's some sort of commercial benefits, one option we've been thinking about is that there be some sort of trust fund set up, and it could be something like a small percentage of the funds that are generated from the exploitation of materials goes back to provide conservation for macadamia or more generally going forward. In situations where there's indigenous peoples involved, it's more complicated. We've been talking with some indigenous groups about the possibility of setting up a collective bodies that work to oversee trust funds. These have existed in Australia and other countries for a while now. There's a number of issues with these, but it's something we've been trying to deal with. I think with that, Ros, I might just stop now.
Yep, that sounds great. No, that's terrific. Thanks very much, Brad. The 20 or so people in the room enjoyed that and appear very engaged. So what we'll do now is, because we had a bit of a late start with some hiccups, we'll just have one question now, I think. Are you going to, I mean, I know it's very late there. Are you planning to stick around for our discussion at the end? It'll be 1 o'clock in the morning.
I'll try my best.
Okay.
I'll try my best.
Yeah, do your best. Get some wine. Mm-hmm. Okay. So let's have a couple of questions for you now, and then we're going to go to Jane after this. So I've got some questions. One question I just had, I'll just see if there's any hands up first. You said something about orphan plants. I don't understand what that is.
Okay, orphan plants, it's a plant that we don't know its parentage or its kinship. So if, let's take the macadamia collections at Alstonville. No one knows where they were, well, let's say, for example, no one knows where they were collected. Those plants, we don't know who it is we go to get permission to get prior informed consent. So we're using the language in copyright law, they would be orphan plants. An orphan work in copyright law is, say, a photograph where you don't know who the photographer is, or you can't find the photographer's son or daughter, whoever it is, who they left the transfer of the copyright to and they died. Is that?
Yep, that answers my question. Any more questions?
Good.
Just a quick one about historical collections of, like, say, wheat and barley. They would be exempt, right? Because they're crops, because they're those important collections, you know, the Russian ones and so on. Is that correct? Are they exempt from these protocols?
They would be exempt because the Nagoya Protocol doesn't apply to those plants or those plants that are in the International Treaty on Plant, the Annex 1, the International Treaty on Plant Genetic Resources. So soybeans, for example, would be caught. But there's still a lot of incredible. Yep, they're not in the treaty.
Don't know if that expression got captured online. Quick, and a question from David. Could we have a microphone here?
Yeah, quick question about.
Can you hear David?
Yeah.
Future-proofing policies for historical data. I mean, we're dealing here with a field of that's, you know, you've seen the graph from Andrew, you know, like it's exponentially growing in terms of the amount of data that's been produced. So in just a few months or a year, we'll have some examples of, you know, genetic studies that will sequence something and then suddenly realize, attribute those orphan samples to something that's actually much clearer where it originated and what's its connection to a country or indigenous group. You see what I mean? So we need to have policies that, of course, historical data is important, but, you know, that can also cope with the fact that being optimistic with this question this time, it's that we're going to see clearer and clearer in the future because we're going to generate more data and more knowledge about those. See what I mean? So.
I'm incredibly skeptical about the capacity of that sequence information to provide the information that is needed. It might tell you there, you know, it is possible using different sort of tests to identify the location of plants in certain contexts. But it's difficult to know who the landowner was at the time. And if something was collected 100 years ago, that person now has, you know, 150 children, you know, grandchildren or grandchildren, because they wouldn't have passed on those rights to anybody in particular. It's impossible to know. Well, the legal system doesn't have the capacity to work out who it is that you negotiate with in that context. So, you know, I think even if you could find out exactly where it came from, it would still be really difficult to get the appropriate permission going forward.
Well, I think this is great. And I think we've had a couple of speakers and they're already disagreeing with each other, which is wonderful. I'm not disagreeing, demonstrating the complexity. So thanks again, Brad. I know it's 11 p.m, so I thank you for being there. And we'll move on to Jane, who is also who you know, who's also online from New York. So let's just thank Brad in the usual way. Now, I think if I just escape this, and I'll just share the screen again. Oh no, well, Jane can come on. That's the best thing.
Hi, can you hear me?
Yeah. Hi, Jane. So,
hi.
So Jane has come to us from New York, where it's not 11 o'clock at night, I can see.
No.
She's originally from New Zealand, but is working in New York. Another person working in the legalities of tracking Indigenous knowledge. So we're running a bit behind because we had a few technical issues. So I'm sorry about that. But we'll pass to you, and we've got coffee break after this, so we should have enough time to have you and have your questions. So thanks. Over to you. If you can just share your screen, then we'll be able to see it.
Yeah, sure. I'll just share my screen. Can you see the screen?
Okay. Yes, that's good. We can see that and we can hear
great. You. Thanks, Roz. I'm really happy to be here. I'm actually from Australia, not New Zealand. So I should just clarify that little part. I'm an associate professor at New York University in Lenape-Hoking, or otherwise known as New York. I'm going to talk to you today just about a project called Local Context, which, following from Brad, deals with questions of data provenance and indigenous rights, particularly in the context of data, but also with historical collections of traditional knowledge. So just a kind of a little bit of a framing around where this project comes from and what it's in response to, kind of thinking about Indigenous data sovereignty. And Indigenous data sovereignty is becoming, you know, one of the most important things to think about in relationship to data sharing, particularly around Indigenous rights in terms of data. And so Indigenous data sovereignty is a discourse that is about rights and interests that comes around from thinking about treaty rights, intellectual property, Indigenous rights and relationship to research ethics and data ethics. Indigenous data sovereignty really is about data for governance and governance of data. So gaining access, so making sure Indigenous people are able to access and use data that comes from their lands and their waters, but also so that they have the capacity to govern that data as well. So managing access to that data to ensure that they can benefit from the use of the data. This project also sits within in relationship to the CARE principles. The CARE principles were developed in 2019, again, aligning interests in Indigenous data governance. And they're complementary to the FAIR principles in the sense that they work alongside them. They're different in the sense that they're not as data-centric as the FAIR principles. They are emphasizing relationships to data, particularly along the lines of collective benefit, authority to control, responsibility, and ethics. And so we kind of see the FAIR principles and the CARE principles and the trust principles as being kind of high-level policy informing not only how repositories are caring for data and thinking about data sharing, but also how researchers are becoming responsible for what happens in relationship to how that data is generated in the first instance. So within the CARE principles, I'm going to talk to you about the notices and the labels. There's lots of ways in which the CARE principles are being operationalized. I'm going to talk to you about two of these, which is the traditional knowledge and biocultural labels and notices. So the problem that this project was really developed to address, and this project has been I guess, in emergence over the last 10 to 14 years, is really thinking about the challenges that Indigenous people have in relationship to finding their collections on the one hand. So every Indigenous community has enormous collections of and data held in archives, museums, libraries and repositories and other online databases around the world. However, significant information about these collections, including proper provenance, is missing. It was just never considered to be important when this material was collected. And you know, from my background, I have an intellectual property background similar to Brad. This is a real problem for the communities that I've been working for because they're not the legal rights holders of any of those collections or data, which really means that issues of responsibility and ownership really continue into the metadata, but it means Indigenous communities have very little say over how that material gets used. As there are more researchers working and collecting information and data and samples from Indigenous communities than ever before, this question of where that material comes from, the actual making sure that proper provenance is connected to that data, continues to be an uphill battle getting that to occur. So if we kind of think about within the relationships of FAIR and CARE, one of the biggest challenges that Indigenous people have is that you can't govern Indigenous data if you can't find Indigenous data. So in the first instance, most Indigenous collections are not FAIR. They're not findable, they're not accessible, they're not interoperable, and they're not reproducible. So this kind of challenge really sits at a foundational level for Indigenous communities. And this is compounded by the fact that Indigenous communities have been overstudied, subject to intensive research and study. And this is just some photos from a collection that I've been dealing with, which is a historical collection from the Lewis and Clark expedition in 1804, 1805 in the United States. These are the plant specimens that were collected in that expedition that are now can be sequenced. And so without the kind of proper information that sits with those plants upon whose lands they were taken, that kind of missing information continues as these plants are being sequenced. So with this kind of intellectual property background and understanding that there was kind of a significant challenge for Indigenous people in relationship to control and governance of their collections, particularly because of the ways in which historically intellectual property law has functioned as a tool of dispossession, we started thinking about what else could you do that could mark those collections differently, could bring Indigenous cultural authority back into the record, back into the catalogues, back into the metadata. And this is where we started thinking about traditional knowledge labels. So the traditional knowledge labels are really human readable and machine readable digital tags that convey particular kinds of information from communities in relationship to provenance, in relationship to cultural protocols, and in relationship to permissions. How they work is that the icon itself remains stable. You can't change that, but every community has the capacity to define what the terms of the label mean. So what attribution means to the community. You can see this is a slide that demonstrates some of the spectrum of the labels. Attribution label is a label that almost every community uses immediately. This is a label that brings communities' names back into the record. But there are also labels that kind of reference certain kinds of cultural specificity or cultural sensitivity that might sit with particular kinds of information, as well as what might be the seasonal relationships to the use of certain kinds of information. This is particularly within a traditional knowledge, cultural heritage context, where certain kinds of knowledge about plants and knowledge about the way in which plants are harvested are connected to particular kinds of songs that might be heard at particular times of the year. So for example, you don't harvest sweetgrass in the winter, you harvest sweetgrass in the summer, and there are particular songs that are sung as sweetgrass is being harvested. So this kind of larger cultural and knowledge ecosystem that sits around a particular kind of plant. And the seasonal label is able to mark that record differently in relationship to it. This is just an example of the customization of the labels that our system allows for. This is an example of the attribution label being customized by a community in what is currently known as British Columbia. This is the Scarlets Band of the Stolo First Nation. And they've customized attribution into their own language, Halkomelem, to be Squix Quas Tek Tumuk, which literally means name and place. And then they include a description of why they're using this label. This is just another example of the labels being utilized within digital publishing contexts. This is a digital publishing book, and the labels are being utilized here to kind of really give readers, giving users of these kind of the digital space, a different kind of understanding of how and what the kind of content of this book is, and how the labels kind of demarcate how that use can be made. This is again another example of the labels being utilised in another context within Australia, which is a Europe Justice Commission, really centering Indigenous data sovereignty and data governance, particularly within the way in which evidence is being collected within this Justice Commission, the first Justice Commission in Australia, to look at histories of colonialism and the impact of colonialism on Aboriginal people within a Victorian context. And they're utilising the labels there. I just want to share this kind of one example, which really demonstrates how the labels get into digital infrastructures, which is really the kind of main point, and then kind of move to the biocultural labels, which are kind of more relevant to this context, but I have to give you the setup for that. This is a record from the Library of Congress from some recordings from 1890. This is a really impoverished record. This is largely what most records look like with Indigenous collections. There's not enough information. Indigenous communities have not been part of these conversations. This is kind of what the record looks like now after having those conversations with the Passamaquoddy community, who've then added 3 of their labels: the attribution label, the outreach label, and the non-commercial label. What this does is kind of creates a different kind of relationship and kind of different kind of authority that sits with these recordings and will now on. And what this has also then prompted is, where do those labels sit? At the moment, they are now sitting within the rights advisory. This is important because those rights have largely excluded Indigenous people. With the labels, those rights are now put back into the record in a different kind of way and are actually added as the first rights. And one would say the kind of persistent rights, the rights that continue and have longevity in relationship to these recordings. But with this, obviously, comes a question about where they start to sit within the Dublin core and also within the Darwin core. For our purposes, and kind of what has become the standard, is that they are sitting alongside, again, within the rights field within Dublin core. And then probably what was the most important part of this project was that this is the JSON field, a new field for traditional knowledge labels was established within the Library of Congress. And what that means is that any Indigenous community that's utilising labels and has collections within the Library of Congress can actually add their authority through the traditional knowledge labels back into one of the biggest institutions in the world. So as we were developing the traditional knowledge labels and recognising that they were really important tools for Indigenous communities, my colleague and I, colleague Maui Hudson in New Zealand from the Whakatohea Nation, we worked to develop the biocultural labels within a kind of a genomic science context, thinking about kind of what would biocultural labels look like. And over the last 4 years, we've really built these out in a different kind of way. These labels speak a little bit more to permissions and to data provenance and less to cultural protocols. Nevertheless, they're bringing Indigenous authority back into the record in a different kind of way. What we advise is that, you know, the traditional knowledge labels were really developed to deal with the legacies of exclusion within historical collections. The biocultural labels are really kind of forward-facing in the sense that they're, we encourage researchers and communities to add these labels as data is being produced. This is just an example of those labels and the traditional knowledge labels and the biocultural labels being utilised within Maui's community, particularly around some genetic sequencing of mussels that are on, they have marine interests in New Zealand, in where Whakatohea Nation is. And they're kind of utilising both the biocultural labels to kind of indicate the different kinds of research use from those genetic sequences, from the mussels, as well as the traditional knowledge labels, because they have particular songs, particular chants around that kind of part of the marine landscape that is also connected to where the science is happening. So connecting traditional knowledge to the different kinds of genetic sequencing that's happening for the mussels. So the labels really communicate 3 key things. They communicate provenance, they communicate protocols, and they communicate permissions. Again, kind of making visible and transparent the different ways in which Indigenous communities want to be involved in research and want to have a say in how their data is utilised into the future. So as we were developing this project, of course, it was kind of focused initially on the Indigenous needs and interests in this space, but also we've kind of understood that researchers and institutions also need particular tools to help them engage responsibly with Indigenous communities and to also disclose Indigenous interests that sit within collections that Indigenous people might not have access to. So we have the kind of open to collaborate notice, the attribution incomplete notice, the biocultural notice, and the traditional knowledge notice. So a researcher might not know what the cultural protocols are in relationship to the kind of research that they're doing on Indigenous lands and waters, but they do know that there is associated traditional knowledge, or they do know that there are biocultural interests. And in this instance, that means that researchers can add those notices. So this is kind of an example of some of the open to collaborate notices that have been put in place at institutions. This is kind of where we suggest that institutions and repositories, as well as researchers, start is to add the open to collaborate notice. This is just being added to different kinds of universities, different kinds of repositories, really kind of signaling an intent to act ethically and act responsibly, and actually to work with communities to bring names back into the record, bring attribution, bring provenance back into the record. This is an example of the Open to Collaborate notice being utilized within a genomic database called Geome. And this has the capacity for, again, researchers to then add associated traditional knowledge and biocultural notices with their data as they're uploading it into this particular database. I don't know if David's going to talk a little bit about this example. This was probably the first biocultural notice added into a vaccinium database. These were sequences that came out of New Zealand, plant and food research, and has been added into the vaccinium database, indicating that there are biocultural interests that Indigenous communities would have with this bilberry sequence. This is another example working with Maine eDNA project, where there's kind of particular samples of soil and water and particular species happening on Maine, kind of working to add particular notices at the very beginning of field research. So these are just kind of some of the developing workflows that we are collaborating with the Maine eDNA team to work out exactly when notices get added to samples, what happens in the post-field sampling within the lab when the notices are added, and then what happens and how the notices are maintained across the different data ecosystems as they move out of the Maine eDNA database into a much larger database like GenBank. So this work is really focused on maintaining provenance across different kinds of data ecosystems. So from communities to institutions, to aggregators, to users, and to publishers. And that includes with different kinds of material, from consultation permits to sample vouchers to collection metadata, really making sure that sharing protocols are part of what the data ecosystems are recognizing. This leads to different kinds of research transparency. We've been working with ORCID. Within our system, the Local Context Hub allows for ORCID to be part of how you register, but also working with ORCID to see how we can kind of pull, if a researcher has added a notice into, in their projects, if they can then display that notice as part of their user profile. And then finally, we're just also working with JSTOR around some of the kind of older historical collections, voucher specimens, in relationship to how you would add notices and then labels to the kind of metadata within JSTOR. So this is just kind of, again, the preferred approach that is coming out of some of the data implementation work that we've been doing. This is with DataCite, kind of working out where the notices and the labels should go, what the different values are for them. Again, they've kind of decided that the best practice is including the notices and the labels within the rights fields. And then just finally, in kind of how the labels and the notices function as particular tools to complement some of the requirements within Nagoya. The biocultural labels in particular kind of think about consent and consent verification, which kind of align with concepts of free, prior, informed consent. The biocultural labels around research use and open to collaborate and open to commercialization really align with frameworks around mutually agreed terms. And then some of the other labels around provenance, multiple communities, and clan authority really pushing towards benefit sharing. If you don't know where the data comes from, you can't connect back to it in terms of kind of creating opportunities for benefit sharing. So again, just kind of why data provenance matters so much within these emerging fields. So I'm just going to finish there. Feel free to contact our local context program lead if you have kind of further questions or want to engage or utilize this system as part of your own work.
Thanks, Jane. That was terrific. Are you able to take, got time to take a couple of questions?
Yes, sure.
Yeah, that'd be great. So we're going to go to a coffee break now, but we'll have a couple of questions first. So we've got time to get the next session set up.
Great.
So I've got some questions from people. I did have a question about, it slightly freaked me out, the thought that eDNA might be captured by this.
Yeah, I mean, it's data provenance. The eDNA team are sampling on Indigenous lands and waters, and those communities want to know, partly because they're really excited about what's going to be found out in relationship to some of the eDNA research that's occurring, but they want to be part of the conversation. And so the team from Maine is really committed to making sure that as many people, and particularly recognising that since the samples are coming from Indigenous lands, that they're part of those conversations, and that's that's connected as part of metadata as that kind of research moves forward. So that team is really excited to be kind of thinking about ethics and responsibility in relationship to Indigenous rights and interests, and to be building that into their workflows from the very kind of first moment of doing field samples.
David has a question. We'll just get the microphone. I was interested that you're working with ORCID, because my thought was, is this something that's going to go the way, you can't publish on genetic sequences now unless you put it in an open database. Mm-hmm. Hence actually all the rest of the discussion we're having. But also I think this is something you could do if you're working on plants that collect on Indigenous land, whatever that means when you're in an invaded country like ours, like Australia, I mean. Then you could have some sort of collaboration with that in the journal. Anyway, it was just a thought bubble while Dave got his microphone.
That was my question also. Yeah, if you had conversations with publishers and editors of journals, and what were the reactions to not enforcing, but at least strongly encouraging the use of biocultural labels for publication?
Yeah. That's a great question, David. Thank you so much. Yes, we have been having increasing conversations with journals around including the labels as part of kind of publishing policy, particularly because it starts to disclose different interests, acts as a form of disclosure of where that data has come from. Certainly not mandated in any way, but does provide different kinds of researcher transparency and accountability in relationship to their data sets and who's been involved with them. Some of our conversations have been with both nature and with molecular ecology in relationship to utilising the notices and the labels as part of researcher tools that are kind of striving towards that different form of accountability and transparency. So yes, I think that we'll see that over the next couple of years really emerge as a new dimension of publishing data that is connected to Indigenous people.
I actually have a cheeky extra question, but do we have any from the audience? just one more. It was just the important thing I thought that Brad raised with the historical collections, and has also been alluded to by Andrew in his and what you said, is that this will at least temporarily may restrict access. And as a scientist, what you want is actually to have lots of access to everything. So do you think if there's going to be some restricted access to materials, this will be temporary, or is it waiting for some kind of legal framework? I understand there's cultural sensitivities and that would be ongoing, but thoughts?
I don't know if it's about restricting access, but making it there being more inclusive access, in the sense that there are people who do have rights and interests that have largely not been at the table having those conversations. And so what the labels do is they kind of bring people to that, that space in a different kind of way, not to necessarily restrict access, though of course there are certain kinds of sensitivities that exist in relationship to particular plants and particular species, and kind of understanding different worldviews in relationship to that is something that I think we really do have to think about seriously. But it's also adding voices that previously weren't present, that also opens up different kinds of understanding and different kinds of ways of it kind of makes for better science, actually. And so I think that what the labels and the notices do is they enable a conversation that has previously not been possible. And from that, we learn new things. And so I think about it less as restricting collections and more as kind of opening up the conversations of who's participating and who's able to be involved in those conversations, and kind of what that means and what we learn from that.
Great. Well, thank you very much. And so we'll just thank you in the usual way.
Thank you, Roz.
We're going to have a coffee break now. After the coffee break, we've got David, I can't pronounce your surname, Chagne, David Chagne, speaking, and he will continue to develop this in the aspect. And then after that, we have allowed some time. There's a little spot there. We're going to have a discussion before they start serving drinks. So that will be an important thing. So thanks, Jane, and thank you everybody for your attention. Thank you. See you back here at 4. Will I stop? Yep. Okay, it's 4:05, so I'm going to start. So thank you for coming back to this session. We have one more talk, and then we have a discussion session at the end with another expert, Rodrigo Sara, who wrote some of those papers that Andrew referred to. And so what I'm going to do, and that'll run for as short or as long as we want it to be, or as formal as informal as we want it to be. So the last talk in this session is David Chagne, and he is from New Zealand, and he is going to talk about some of the work they've been doing with research with and for Indigenous communities, and our European scientists' perspective. So over to David, and we'll hear from you. Thanks.
Thank you, Ross. Thank you all for coming back from a cup of tea. So today I'll present about some of my experiences and perspective working with Maori communities as an illustration of how, you can do genomics research in plants and animals in partnership and for indigenous peoples. So just to introduce myself, you will hear from my accent. I was born here in France, actually. So I'm probably the most indigenous person here in this room today, and moved in New Zealand, Aotearoa, back in 2004. As many of you, I'm passionate about science, understanding the world around us, and be fair to people while doing this. So I'm not a lawyer, I'm not a policymaker, I'm not in that field. I'm actually a practitioner. I do biology, I do genomics across many species. And among those species, my group focuses on are native plant and animal species, endemic or native to New Zealand. And I'm from Plant and Food Research, which is a research institute that actually works with the horticulture, aquaculture, and agriculture industry. So a lot of the species that we work with actually have some commercial values in addition of being important culturally to Maori people. So I'll start my talk. I won't talk much about Nagoya because, as a matter of fact, New Zealand is not a signatory of the Nagoya Protocol. However, it's got quite a peculiar situation that's due to the Treaty of Independence of New Zealand from 1840 that was signed between the English Crown and Maori chiefs from around the country. And this document is very, very interesting because first there's two versions of it, one in English and one in Te Reo Māori, in the native language of Maori people. And there's some key concepts that are addressed in the second article of the Treaty of Waitangi. The first one, there's a word that I highlighted in red here, that's the term taonga, which is actually means in Maori something that's treasured, that's precious, you know, something that the indigenous people of New Zealand have, you know, a strong attachment with. But it's also talking about other notions such as tino ranga tiratanga, which is, you know, recognizing that the crown of the Queen of England recognized an absolute sovereignty of those treasures to Maori people, and that the Maori people had a right of kaitiakitanga, which is, you know, guardianship of those species. Okay. Unfortunately, this treaty was a little bit buried and forgotten for many years. It was, you know, like some piece of land and some areas that were taken unduly by the Crown of England. But recently, through the Tribunal of Waitangi, the Maori entities, you know, tribes and sub-tribes, can now have the opportunity to reclaim those lands and assets that were unduly taken by the Crown. Okay, so now we've got a situation where Maori people can, in many examples, will own large tract of land with some species growing on it, or own some fishing quota and so on. So suddenly we've got a situation in the last 20 years where more and more Maori tribes and or super tribes, or depend on level they went to the tribunal with, but they now have strong resources and they're well organized actually to take advantage and apply the guardianship and the sovereignty of their treasures. So at Plant and Food Research, we've thought about it. You know, we've got a history of working with some horticultural plants, and we thought, you know, if we want to be in the business of working with native species, you know, we need to develop a strategy and a bit of a set of guidelines how to work with those type of species with Maori people. So I'll give some examples, and I've organized it in 3 different parts. You know, one part talking about what we do in terms of engagement with communities. Another one about partnership, and this part will be a lot about some of the key aspects of the Nagoya Protocol, which is about benefit sharing. And also the last part will be, you know, how to protect the indigenous flora and fauna, and how to also protect Maotaranga Maori, the traditional knowledge that Maori people accumulated over the many years they've been in Aotearoa. So in terms of engagement, we've heard about it in previous talk, and that's part of the Nagoya Protocol also, but we recognize that the key principle is that we need to receive informed consent from the rightful Maori guardians before doing any work on biological material, or before doing any work on DNA data that we may have produced or that we would like to reuse. What's really key, and I think Brad mentioned that, is that in many cases, a critical part of the process of engagement is actually identifying who are the rightful people to engage with. So basically, you don't go in the streets and talk to the first Maori person that you see and say, well, what do you think about our research? Would you approve that? That's not actually the way to do that. You know, you need to find the right people from the ancestral lands where those plant or animals have originated, and talk to the hierarchy and the people in those entities, whether it's a tribe or a super tribe, or sometimes just at the family level. But we talked about digital sequencing information, but there's also a lot of metadata, including traditional knowledge, that actually add a lot of value to those digital sequencing information. And this is, you know, traditional knowledge that needs to be also protected, and any use of it needs to be consented. And what's really important is that it's not only about, you know, getting a an informal consent to the research, but we need to be able to prove, and demonstrate that those consents have been given, and it's recommended to use written consent. So I'll give some examples of the work that we've done. Probably 8 years ago, we initiated the genome sequencing of a species native to New Zealand. It's called Manuka. And a couple of years ago, we sequenced the hoki genome, which is a deep water fish species that you find all around the Pacific. There's a lot of it in New Zealand, Australia, but also some around the South American coast. So we've developed genome analysis of those species. And basically, instead of dumping this into the usual NCBI type of databases, we actually deposited them in a New Zealand-based repository. So physically, the data is hosted into a server that's actually in New Zealand. And the access is not, it's not a pay-for access. It's not, a private access in terms of, what you would have if you were dealing with something with a commercial interest, but it's managed. It's not totally open access. So basically, when you go through that Genomics Aotearoa data repository, there's a small form to fill to ask a few basic questions about the whys. Why are you doing this research, and what are the benefits, the expected benefits to Maori communities? They are scary questions. They're not easy to answer. I mean, it's not as easy as clicking a link to an open access database and get your data automatically. It makes you think about it, but I think it's extremely important. So in term of the Manuka genome raw data and analyzed data, we identified the local tribe where we grew the trees that were used for doing the DNA extraction and the DNA sequencing. The local tribe, Rangitane or Manawatu, are the people who are the guardians of this data. And they actually receive those requests. And in a very timely manner, I can tell you, they just give a yes or no. Usually it's a yes for people to get access and reuse this data. In terms of the hoki, that was a bit more complicated because hoki, as I said, is a fish species. Well, they swim all over the place. And it's very hard to say, that fish is coming from that particular ancestral land, obviously. So in this case, we've engaged with an entity called Tehuhu Kaimoana, who's an entity that actually coordinates all the fishing quota held by 58 different tribes of Maori around New Zealand. And they are the ones speaking to those 58 iwi, those tribes, directly, and giving the yes or no and the conditions for reuse of the genome data. All right, another example of what we've done in terms of recognizing the guardianship of Maori over the data, and that points to some of the things that Brad said earlier in his talk, is about best practices. And in this case, I'm pretty sure Jane showed an example also of those sort of what happens to the data and where do you attach the biocultural labels that you presented about at each stage, whether it's to the publication, the raw data into a database archive and so on. But what we've done is that we've talked to, in this case, it was Tehuhu Kaimoana, so that this entity of Maori people coordinating the fishing quota for 58 different tribes. And we sat around the table a few times and developed a very, very specific taonga, so treasured entity data cycle that asks, the question at every step of a project, what sort of questions do we need to ask and answer to go to the next step. So it's not only about, data generation and data analysis, but it's recognizing that, you have to start with the top part over there is Wananga, having a very initial type of meeting saying, well, what's the goal of the project? To all the different steps, including collection of samples, data generation, the analysis, but also what's going to happen in the future. Once your publication, your initial goal is achieved, this data set will have probably a very likely to be reused for another purpose. So what are the conditions of reuse and how do we reconsent at every cycle? So this is what we've put in place, and we've done that that was co-designed and co-developed with our Maori partners. Another type of tool that I won't go further into details, but I just wanted to put a slide about it because I'm really enthusiastic about it. This is those biocultural labels, because if you have perpetual metadata fields that can stick to your digital sequence information, that solves a lot of problem. You know, you can keep that attribution to those community. You know who are those guardians you need to talk to and what are the conditions of reuse. That's a very, I think, a very useful tool to have. All right, so now I'll talk about, after the part about engagement, I'll talk about the parts about partnership, and just, you know, refresh you about a few of the things in the appendix of the Nagoya Protocol, some of the examples given, you know, what do we mean by benefit sharing? So of course, it could be some monetary type of benefit sharing, but there's a lot of other examples that are non-monetary and worth considering. So I'll give some examples about that. So this is what we wrote in terms of our guidelines and policy, my institute, in terms of the Maori context, you know, and we recognize that we need to share, you know, a reasonable proportion of any benefits that we've got. So basically, an institution like mine, who makes, you know, a lot of revenue out of plant varieties and royalties, we recognize that in this case, this is very different from dealing with a kiwi fruit or an apple industry. We'll have to deal with a long-term, you know, indigenous community that will have a very different collective way of apprehending ownership instead of having a corporate way of dealing with ownership. And that brings some totally new concepts that we haven't quite solved yet. But it means that we're ready to have those sort of very difficult discussions. So I'll show some examples. Again, I'll show the example of Manuka, which I mentioned before. So it's a native tree of New Zealand, and you find some in Australia also. It's the basis of quite a fast-growing honey industry. It's very high-quality honey with very unique antimicrobial properties. And interestingly, a lot of the lands that were retrieved by Maori through the tribunal of Waitangi, you know, like they were taken from them by the crown, given back by that tribunal, they're like not really good for horticulture or any other type of agriculture. And a lot of it was covered in that horrible bush that 20 years ago had absolutely no value and was, you know, basically sprayed just to get rid of it. But they realized, some researchers realized that the quality of that honey, and suddenly the Maori realized they've got that gold mine. They're sitting on a gold mine of unique plant material that gives that very high value end product that can be sold all around the world. So those Maori groups were actually quite started to coordinate between themselves, and some of the, you know, the beekeepers, the honey companies owned by Maori and so on, started to coordinate into a more a unified sector. And they came to us and said, well, can you help us ask quite a few key questions for us? You know, what makes that honey, for example, unique to our ancestral land? How is it different from the Australians, for example? And that's the question they ask. And we put together with those Maori groups, we put some funding applications, we got some funding, and those Maori groups were actually governing the project. So they were actually in charge of all the decisions about what to do with the data, how to approach the different landowners where we would get samples for our analysis, and asking very difficult questions at the onset of the project about how we're going to publish this. And I can tell you the very first meeting was a 4-hour meeting talking about the publication that would come out of the project and how we disagreed about what we could say and shouldn't say and so on. But it was fascinating discussions that were, and we ended up, you know, meeting in the middle and having something that was beneficial for everyone, not only the scientists, but also for the communities and the Maori people. So some of the knowledge that we developed, you know, through this analysis was quite an exciting project. You know, I could talk about it for 45 minutes, but I won't. We understood, you know, what's the genetic diversity of Manuka across its natural range. And what's really exciting is that there is a strong cultural connection between the plant and animals growing on ancestral land and the Maori people. Pretty much you could define it as co-ancestry. A Maori person would consider that a Manuka tree or a hockey fish growing in their ancestral land is from the same family. So this means that, you know, it starts to ethically, you know, if you take a sample, a DNA sample, some digital sequence information. It means that any reuse of this data is like, any ethic approval that you would need to do for doing, human pharmacology type of experiments, okay? You need to reconsent and so on. So it's exactly the same, that those samples are part of the family, okay? But fortunately, also, see if you flip it, if you look at the genetic diversity and demonstrate the uniqueness of Manuka across the country, and if luckily it matches the ancestral land, that strengthens actually that notion of whakapapa, of co-ancestry between human and the plants growing on the ancestral land. So the Maori were on the moon by this type of research that we've done, because it was actually strengthening a long traditional, a core concept in their worldviews. And also what's really important is that if you know about the genetic diversity of a species, then suddenly you've got better, more informed tools to actually be the rightful guardians and the careful guardians of that resource. So for them, they're very keen on being those guardians, and this data has contributed. Not only this, other aspects, traditional knowledge is important also, but the DNA sequencing information we got was very important for them to exercise that key principle of guardianship, of kaitianga, that's part of the Treaty of Waitangi. So very, very important to them also in this aspect. So I'll talk about some of the shared benefits in this project. So they were like co-designing, governing the project, but also, the results we got was important for them to demonstrate the uniqueness of each type of Manuka. It means that now they can commercialize some of their honeys based on its provenance, like French wines. So it's like a terroir type of situation where you can make a premium product, not only by its quality, but where it's coming from. But also we ended up with some Maori people being co-authors of one of the papers that we published about that genetic analysis of Manuka. And that's very important to recognize, their role across different aspects. So one of them was doing data crunching with me. There's another one was doing DNA extraction. Kiri was doing the coordination of the sampling, and Victor was one of the governing people of the project. So they were all part of that paper. And along the way also, in terms of benefit sharing, we've actually employed Maori students and Maori field technicians to do the sampling for our DNA analysis. So we contributed to the social well-being of those communities that, as a matter of fact, some of them were very high unemployment rates. We employed 4 of those people in those areas to actually support them for summer, to do some work, do some research. And in a way that got them into the business of being interested into science. So we got 2 Maori students that now, they increase that gap that we've got, decrease the gap that we've got between, representation of indigenous people in our profession. There's very few indigenous people being, researchers in general. So it was important for us also to contribute to that, to bridge that gap. So on that topic, over the last few years, Jane mentioned Maui Hudson in our previous talk, and what we've done in New Zealand, and that's copied to a system that was developed in the US, is that we've developed a summer institute of indigenous genomics, where every year for a week we take 20 Maori interns, budding scientists, and take them for an entire week to talk about not only technical aspect, but a lot of ethical and cultural aspects of genomics in general. And that's extremely successful. We have 5 years of it. We had an international conference of indigenous genomics with people coming from various parts, like from First Nations in Americas, Australia, and South America. That was fantastic. Really, really great meeting. And now there's a Sing Australia that exists. It's gone for 1 year. And we, once in a while, we send some of our interns from New Zealand to the Sing USA or Sing Canada. All right, so now I'll talk about Protect. And one key aspect is that you might have noticed that through my talk, I've been using some Maori terms, and that's actually quite important. The language, Te Reo Maori, is actually a treasure also. So any use of the terms that I use in terms, using the term for kaitia kitanga or whakapapa, those key cultural values need to be respectful and genuine. Okay, so that's very important from us. Scientists that aren't of Maori descent to use it in a very genuine way. We've had some difficult conversation also about, the impact of new technologies. Some of my colleagues are expert in gene editing, plant transformation. What could be the impact of using gene editing in species native to New Zealand, or even in species that aren't native to New Zealand, but would have, an impact to Maori communities. And there's been some really, really exciting discussions that things aren't black and white. In fact, it's pretty nuanced. And there's some, I'm quite optimistic that there would be some way to work with Maori on those challenging new breeding technologies. I'll go quicker on this slide because I'm conscious of the time. But really, what's key here, I underline the term, being a trusted custodian. Well, you've heard about a FAIR principle for data practice, in science, and basically a key aspect of the FAIR principle is that, if you want your data to be findable and so on, is that you don't own the data. It's not because you've pressed a button on a sequence machine that you own that data, okay? And all we are as a research institution is just a bunch of lucky people who have a supercomputer that can actually hold the data and have the knowledge to crunch this data, okay? We're just trusted custodians. We're not the owners of those data sets. And that's quite a key aspect to address. All right, so in terms of protect, I'll give another example. This is an example with the local tribe I mentioned then earlier, the Rangitane of Manawatu. And this is an example of, sitting in a room with them again and saying, like, what are the questions that you're very passionate about? And one of the species that we've got in our region is probably less than 50 trees, adult trees living in New Zealand left. It's swamp mire, so it's a species that grows in swamp, which have degraded a lot. And on top of that, it's highly susceptible to a fungus disease, myrtle rust. So we've talked to those and they say, well, we need to replant and protect those. So can you use some genomics type of tools to tell us, how to maintain the population to a healthy level in the future, how to replant, where to take the seeds from, what are the best mother trees and so on. So that was quite exciting. And that was 2 weeks ago, I got an email from Paul, who's the guy wearing that orange shirt on the right over there, pointing to some trees to us with his with all his equipment. Well, he asked those very, very pertinent questions. You can read the emails, but very, very pointy questions about what do we know? We've got those trees here. Should we use this one and so on? And those conversations, I can tell you, it's my perspective and experience. It's so enriching as a human being and a scientist, to be part of this and co-designing and doing the research together with some people who have skin in the game. They share that ancestry with those species. So we're doing some sequencing. We like doing this and generating pretty much sequencing information from all the adult trees that we could find and many seedlings that we grew. And that's a project that actually, once again, funded by the government to the tribe, to Rangitane, and we're subcontracting for them. So they own it. And some of the activities that we will do with Rangitane is actually, they're going to come and I think beginning of August, I'm sending them in my lab with my lab technician to take some videos of how we do for processing DNA. It's quite exciting, I think. So a lot of public outreach also is part of this process. And another example that's more commercial is working with a group from the same area that Jane showed before, where it was the mussels. There's also that species of fish, kingfish. And we've been asked by the community to actually develop a breeding program, a selective breeding program for that species. So here, there's going to be some IP generated with strong commercial value. And that's, we're starting the first objective of our project with them is actually to get a framework to understand how we're going to share the benefits in the future, as in, like royalties from, high quality brood stocks from those fish. So that's going to be interesting. Not done yet, but it's going to be, I'm looking forward for it. So that's my conclusion. I showed 3 parts, some examples about engagement, partnership, and how to co-protect species with Maori. Along the way, my institute has developed a high-level strategy to achieve those goals. And that involves, building capability and capacity within our organization for Maori researchers within our organization, but also for people like me who aren't Maori, to get a better understanding of how Maori work, what's the worldviews and what to do, what not to do. how to do research in partnership with Maori, but also how to enable, those commercial partnerships in a long term, and in a way that we can share the benefits with those community while respecting that key concept of guardianship for those species. All right, so I've got a few people to thank, but I'll because for an interest of time, I'll just thank you for your interest, and I'll take any question. Thank you.
Thank you. From personal experience, that yellow-tailed kingfish is just fabulous eating.
Yeah.
That's really good. So thanks very much, David. So let's have a couple of questions now, and then we'll open it up to the wider discussion. Andrew.
No, it's not Andrew, but
It's at the front, sorry.
Thanks. That was a really interesting talk, and I think based on our earlier discussion, I personally am absolutely find it really exciting to hear about these sorts of projects, and I think that they're the natural complement to the centralized open databases, and in my view, we have to support both personally. The question I have is about the and this is a boring question, but I've worked in the publishing world as well, and so financial sustainability is always the cost for maintaining this separate database that you've created. Is that something that you bear responsibility for in terms of genomics? And is that something you're concerned about in the long run? In terms of, in general, one assumes that the more complicated the database is and the more controlled access steps, the more expensive it is to run. So I guess that's kind of interested to hear your comments on what that means for you in terms of running it sustainably. Yes, and that's a good question. And we've got those discussions right now with the funding body, the government funding body, who's funding Genomics Aotearoa. That's going for another 2 years, and then we hope for renew, and whether or not we get another 7 years or 8 years of funding, we don't know. But that's a really good question. I mean, we've had discussions also with not only the government, but with the archive type of people of New Zealand, like could they be actually the people ending up being the custodian of this database? I think the key for us was really, we're conscious about the perenniality of that sort of database, but what's also very important is to build the guidelines, to build the infrastructure to make it possible first. And then we'll worry about finding funding in the future. We'll make it work. It's hosted in one of the universities of New Zealand, University of Otago. And I'm sure, if it's not the government supporting it, we'll find a way to get that university to push it. But really the key was, it took 5 years of the project to get to a stage where we've got now a database that's functional. There was a lot of engagement and discussions to what we could do and what would be the shape of it and so on. And I think that's I'll be very optimistic. That's a big achievement to get to that stage already. So we'll worry about the future funding soon, believe me. Yeah.
Yes, at the back.
Yeah, so I guess like the question right now is also about funding, but more from the perspective of like, how do you actually go about it if you aren't local? So, I mean, like you have given like some very good examples of collaboration between researchers and, well, in this case, the indigenous Maori people. But like, how would you, for example, or like in your experience, is it common for people from other countries with a grant to be able to write significant amounts of funding for collaboration with indigenous people into that grant? Like, is that actually something that you might reasonably expect to do? So for example, you apply for, say, $500,000 for a project, and then you want to spend like half of it on you and on your lab and a postdoc, and you explicitly want to offer the other half for like capacity building where you're going. Is that actually something that is feasible in the current funding environment?
Well, if someone from overseas comes with its own funding to do some research of one of those Taonga native species, we would expect actually them to have some sort of engagement. Actually, there was an example. I'll give one. The Sanger Institute, about 3-4 years ago, was celebrating its 50 years. Oops, sorry, but a big landmark anniversary. And they had a booth at the Plant and Animal Genome Conference in San Diego when I went to that booth, and they were showing, like, oh, for our birthday, it was probably 20th birthday, we're going to do 20 species iconic to the UK. We're going to sequence them for free and give it to the community and so on. And one of those species was actually a worm that's native and endemic to New Zealand, and that's, a pest in England. And I said, well, this is good. I understand what you're trying to achieve. This is great, like just eradicate it, no problem. But you realize this could be considered, that you need to talk to Māori people because this is native to New Zealand. And they were like, oh, that's interesting. And they ended up doing it. So this is possible that, people with deep pockets like Sanger would go and decide to sequence something that's actually native to New Zealand and engage with the appropriate people. Unfortunately, it hasn't been done enough in the past. And that's, it's, as scientists, we've got to deal with that now, like the fact that the kiwi genome, as the bird, not the fruit, was sequenced by a group that's not from New Zealand. There's no problem about that. We're not putting this framework so we sequence everything in New Zealand. It's not because we want the absolute monopoly on that, but there was no consent. They used birds, from a museum or some other collections, and that's trying to get around the rules to try to like avoid doing this type of engagement and benefit sharing, and that's not right. So, but if someone has got the willingness to do the type of things we discussed about today, it can be someone from France, Germany, US, China, wherever. It is no issue at all.
Yeah, so I guess I'll just go on since nobody else.
Oh no, I think what we might do, we will go on, but I think we might pull this part of the thing together. And so thank you, David, for that presentation, and we'll get you back to answer some questions if we need to. Yep. So, and of course, thank you formally to all the speakers so far in the session, but we have one more part to go. And I'd like to welcome to the podium Rodrigo, Sarah, and I'll stick around here too. And in this session, I might even, it doesn't matter. We can close that. What I want to do is to, so Rodrigo comes from a background from, I think, working with CGIRs, which I think is very interesting. We have this debate about whether you have individual negotiations between partner organisations. Part of that's what's happening with the Indigenous people. I also work with Indigenous people in Australia and the traditional custodians of various plants. And that's, to me, it's like getting animal ethics rights. We kind of accept those things. So how complicated is it? Obviously, multilateral agreements are simpler to execute and for the databases. So, Rodrigo, I'd just like to hand over to you. Maybe you want to just throw out a few questions and ask people what they think, and then we'll just see. So this is all going to contribute to our working paper. So thank you very much for helping us with that.
How much time do we have before?
I think we're about 20 minutes.
Okay.
I'm sure people would be happy with 15, but let's just do it.
Thanks, everyone. Pleasure to be here, and thank you to the organizers for the invitation. I think you've been treated to a really unique set of perspectives. It's rare to move from a scientific perspective on the use of genomic sequence data and with an open kind of perspective, and then transitioned into traditional knowledge and working with communities, the more TK and IPLC type discussion. And so therein lies the tension, but also the solutions, right? So this whole area is about tensions. It's tensions between what is or should be part of the commons versus what should be under sovereign control. And is that the sovereign control of states, or is it the sovereign control of the individual communities that are stewards and guardians of particular resources? And so there is ultimately a tension between an open
system and a closed system. And the key coming into these international discussions where something will be decided later this year is can some compromise be found that is workable? Can a workable compromise be found that actually continues to meet the needs of the scientific community who have had now 30 years of open databases and being able to access digital sequence information in its many forms readily without friction? Is there a compromise that can be found that is able to honor the sovereign control of states and IPLCs, communities, but actually generate benefits for them in a way that sits comfortably with open science? And that's, so far, few models have been put forward that might actually be able to deliver us a solution. And I guess in an oversimplified way, what we do know is that the Nagoya Protocol, and the CBD, they had very legitimate objectives, but after 30 years of their implementation, the question is, have they achieved what they set out to achieve? And looking specifically at Nagoya, has the bilateral system achieved what it set out to achieve? And can any bilateral system achieve those aims? Is there something inherently limiting in a bilateral system? Is an open question. And so it's if you over-regulate access, if you make it really difficult, either legally or even just creating frictions. I mean, these licenses are incredible tools that have been developed, whether it's Creative Commons, whether it's these biocultural labels or traditional knowledge labels. They're incredible tools for responsible research, but they are frictions for every researcher, and they imply that every researcher can quickly come to terms with what are ultimately legal tools, or they have the in-house support in their institutions to be able to use those tools appropriately and to be responsible. So if you over-regulate access, you reduce the amount of researchers using those materials. Therefore, you minimize the benefits, so there's less to share. So you're actually, by over-regulating access, do you risk that you actually don't find a solution to this ABS tension, that you do enhance responsible research, but you don't provide benefits or enhance benefit sharing? And the other way, if you make access frictionless, the way it currently is now, you create lots of benefits. We have trillion-dollar industries associated with the use of biodiversity, and yet those benefits aren't being shared. They're not enhancing the bioeconomies of the countries from whom those genetic resources are derived, where they originate, or the communities that are the historical custodians. So that's just the summary of why this is such a tricky area, and how do you actually find a compromise? And so my question to each of the speakers is, do you foresee any multilateral model, right? So a multilateral model underwrites access. Do you foresee any multilateral model that could be acceptable to developing countries or to indigenous people and local communities? Or is it simply inconsistent with the degree of control that they need to exert? And that's the answer. Doesn't have to be yes, it doesn't have to be no, but I'm curious, it's whatever you feel it is, but just your perspectives on whether there is there a sweet spot where an equilibrium can be found, or is it unlikely to occur?
So I think we'll take in turn. Andrew, do you want to speak to that first?
Well, so
just here's a microphone coming just in case we are recording. We can delete this if you want.
I thought that actually David's talk and my talk, sets up a nice dichotomy there. Because what I would like, I think that a multilateral system is better suited to generate, in the end, financial benefits in the long run. And I would like to see a situation where indigenous communities have the power and the support to create, the cool kind of systems that David is describing. But then also they have this tension to say, well, if we put it into the public system and let go, we'll get these benefits. So there's a tension between there are going to be where people are getting rewards for sharing openly in the big public, which has huge benefits, as we know. And I think that a multilateral system there can generate money. but it may not generate the same sort of control. And so I would like to see people choosing between those sort of excellent opportunities rather than us trying to break down the public systems into a lot of smaller things, which I don't think will generate. That's my view.
David, we have your response to that.
Oh, is it a tennis match?
You don't have to.
No, no, just, you know, in an ideal world, absolutely, if everything was open, you would see benefit to everyone, and it's, you know, it trickles down. It's not a reality, and in human genomics, there's a massive gap in the benefits of human genomics to indigenous communities. I've seen stats from Australia where, you know, some places have, you know, 10 years life expectancy less for some conditions that have not more, that way, that, you know, more, and that can be treated by, you know, pharmacogenomics, and that's because, you know, well, the panels that we use for human genetics are, you know, overrepresented in Eurasian types and totally underrepresented in other indigenous communities, and that's the history we're dealing with. There's a lack of trust, and because of that lack of trust, it's going to take time actually to get to a stage where we're going to be saying hand on heart, oh yeah, of course, you know, put everything in the open, and everyone will be happy, and every type of community will benefit. This, unfortunately, this is not the situation, and history or the current situations demonstrate that, you know, indigenous communities don't get the share of the pie that they should. So that, I mean, yeah, I'm all for open access, you know, don't get me wrong at all. It's just when you talk to those people, they get hammered all the time, and that's, they lost trust. That's the issue.
Yeah, can I add to that too? So I think that possibly what's not appreciated by people that don't live in a country where, as Europeans, we came and invaded and took over their land, and it's a much worse case in Australia. There is a real lack of trust, and building those bilateral agreements and so on is terribly important. But my question is, and we haven't got Jane here, that if you put these labels on, and I know, you know, you have hesitations about labelling things just because of the cost and inhibition, but if you had a label on something that went with the data, it could be on an open database, couldn't it? And have that tag on it that said this, so that if someone pulls that sequence out, then it just automatically alerts them to something. Is that a possibility?
I believe so. I think it's a good solution, especially some of those tags or labels are clear about potential for commercialization. So it's not that it's closed for it, you know, it just flags that, well, just come to us. So yeah, so yes is the answer to the question, to my view.
Andrew, and then please, others, please comment too. I'll come back to you at the back.
And so I'm finding it useful to disagree with you, David, although I think that actually I agree with you on exactly what you're saying. But so I think that I like the idea of having separate repositories that have different terms of use. I don't like the idea of putting legal licenses on individual sequences, because I think as a researcher, if I go and I download 10,000 sequences, which for the type of research I would do would be a small analysis, and then I have to go through each one and try to figure out what do I have to do, that I'm just going to throw out the sequences, or I'm not going to abide. And that's actually what we've seen, I think, within the publishing realm, is that the Creative Commons non-commercial licenses have their place, but they don't drive commercial benefits back to researchers who choose them. So it gives a false sense of power when you're really talking about money. And so I think that you need to think on the database level, because the individual, now adding biocultural labels because metadata is good, because people it gives them the power to respect that history and that provenance. I'm all in favor of better metadata, always good. It's that trying to put legal benefit sharing requirements on individual sequences, I think has challenging aspects to it.
So we don't have Brad and we don't have Jane.
No, okay, sorry. Jane's not there on that. Is it? Of course, we do have Isabel as well. Sorry, in the green T-shirt at the front. Yes. I don't know if that's green. It's on colorblind. This one.
So clearly I'm an outsider of the discussion, right? But I have a question regarding the role of the governments in different countries regarding this discussion, and if they participate actively in this discussion. For instance, I'm from South America, and I think this is great what's happening in New Zealand and Australia. I think we are really far away from these cases. I don't know if you have any experience on that, or what I'm saying is that is government making policies related with these issues, and they are actively participating?
No, and in the case of New Zealand, they can't, because it's, if the government develops a policy, it's going to be seen as a continuation of colonialism. So you can't have. So ideally, yes, but this is not actually governments. Even us as scientists, because we're funded by government, we've got a clear conflict of interest that we need to declare with Maori every time. And anything that we say is always taken with a big pinch, like a big bag of salt, because we are funded long-term by the government, which is, has taken land from Maori for more than a century. So no, so yeah, it's not simple like that. Yeah. Okay. There's some issues also of indigenous boundaries don't often match political boundaries also. I'm getting a little bit out of my depth here, but I mean, we've worked with the Sami people from Northern Europe for the Pilbara genome, and Sami people go from Russia to Norway. So it's you're not dealing with one government, you're dealing with governments that probably wouldn't talk to each other right now. So yeah.
All right. Thank you.
Just a quick, so the countries are very involved in the negotiations under the Convention on Biological Diversity in terms of sovereign rights. So nations want benefit sharing. The question of how well indigenous peoples is that's a separate. But so nations want because they have a sovereign right for benefit sharing based on national rights. And South America in general actually has a relatively distinct position within those. The voices from South America are very diverse, but there is an overall that South American countries tend to be arguing for some sort of individual tracking of sequences, whereas African nations tend to be arguing for some sort of basically global tax. That's all that is grossly oversimplified, but these are very, very tense, and Rodrigo can speak to this.
I think I just want to highlight that, and I'm an advocate for indigenous data sovereignty. I'm a co-author of the CARE principles, but my biggest concern is that we're currently in the worst of both worlds. Right? Since Nagoya, the amount of collection activity has decreased significantly. The onus on researchers, all the burden of Nagoya compliance falls on academic institutions and public institutions. And it's having a real impact on research. And it's incentivizing researchers. Data wants to be free, and it's incentivizing researchers to actually use materials where there's a path of least resistance. So the current system it's the worst of both worlds, because it's affecting access. And then it's not just, and because there's limited access, there's lots of benefits, but not benefits that are being shared. So how do we shift away from that? How do we find an equilibrium that actually maximizes access and then maximizes benefit sharing? And that's where some new approaches are required. So I believe in responsible research and innovation, in respecting indigenous data sovereignty, but surely the answer for DSI cannot be, let's take this bilateral Nagoya system that has failed for 30 years to generate benefits or facilitate access, and now put that system over the public database system that for 30 years has been quite successful in spurring innovation. Surely the solution is something new and something novel. What I sincerely hope is that solution is not that we create some kind of commons for certain data, with exceptions that are over-regulated to ensure that sovereign rights are respected, because my fear is that that will lead to that fragmentation will lead to a disincentive to use those data sets that are not part of the broader commons, unless you're in a very niche field of ethnobotany, or that you have a real reason to be working with genetic resources from a particular community. But we know genes are promiscuous. We know that metabolic pathways are not conserved. And so what you're seeing in one location, in one species, may be found in another. So my sincere hope is that we can find other solutions. Under a multilateral system, is there a governance arrangement that might be acceptable to IPLCs? Could you build in benefit sharing, both monetary and non-monetary, in such a way as to be attractive for them to have trust to pull their genetic resources into that system with confidence that there will be benefits flowing, and that if benefits aren't flowing, that either there's a revision of the system or they're entitled to take their resources back. So not something that creates fragmentation, but something that creates integration. So these are new, and that would require some kind of new and novel approach that would need to overcome lots of historical distrust and would need to be able to promise significant benefits to have any chance of going forward. And so that it doesn't exist yet, but one of the reasons it doesn't exist is because you don't have the stakeholders in the room. When you hear a researcher talking about how important it is to preserve open data and a commons for DSI, it sounds very logical. When you listen to IPLC advocates and stakeholders talking about why it's so important for historical reasons and otherwise to preserve indigenous data sovereignty, it sounds logical. When you hear developing countries talking about why they need a way to secure and enhance their bioeconomy, it sounds logical. So how do we lock these people in a room and actually have them come up with a solution that is integrative and represents compromise, something new that hasn't been done before? That's what I would hope is an outcome. But this particular treaty compromise will be negotiated by the end of this year, and the fear is that in the 11th hour negotiations that tend to take place, maybe there will be a solution found, but maybe it will again be a solution that doesn't really meet the needs of any stakeholders. As you could argue, Nagoya, the experience 30 years after implementing the CBD and 14 odd years after implementing, or 12 odd years after Nagoya becoming effective, hasn't quite delivered. How do we prevent ourselves from coming up with a solution that is future-proof, that we don't spend another 5-6 years dealing with the unintended consequences of fragmentation or of integration that maybe that doesn't deliver benefits, as the International Plant Treaty showed. But I think that's it for time.
Yeah, so I think that is it for time. I mean, I think it's fantastic. Thank you very much, Rodrigo, for your contribution. I didn't even introduce him properly, as given what you said. He's originally from Chile and studied in Australia and now works in France. So that's, I think you're reasonably transcontinental with it. And special thanks to Isabel Mendoza over here. She's going to wave her hand. She's the Chief Communications Officer of the Global Plant Council. The Global Plant Council is here to serve you and to your society. We will tweet things that you tweet. We have the plant science jobs on Friday nights. There's last Friday of the month. Yeah, every month. Every single month. She even did it on New Year's Eve, which I just thought was a bit much. She does a plant science jobs that you can go on to Twitter, and there's a Twitter storm, and there's various blogs, and every day there's plant science news. So I encourage you to follow the Global Plant Council either on Facebook, Insta, LinkedIn, Twitter. Twitter's the main one, but there's all the others there. So with that, I just want to thank Isabel for organising this session. Thank our tech people who sorted out the Zoom, and thank all of you for contributing, and especially thank you to our speakers and to the ones who are on the other side of the world. So let's get, thank you everybody for coming, and let's go and have a bit of a relax and look at some things and get something to eat and drink.